Enzymes catalyse reactions that affect metabolism at both a cellular and a whole organism level. These reactions can be both anabolic, which are chemical reactions where simple molecules are combined to form larger and more complex molecules, or they can be catabolic, and these are the chemical reactions where complex molecules are broken down to form more simple molecules. The combination of both of these anabolic and catabolic reactions leads to the metabolic reactions that we need for life. These reactions can either occur intracellularly, such as the example of catalase, which catalyses the reaction and breakdown of hydrogen peroxide into water and oxygen, or they can occur extracellularly. Two examples of these are trypsin, which breaks down proteins, and amylase, which hydrolyzes starch down into sugars. For two molecules to react, they need to collide successfully, and there are two things that will affect this outcome: whether they have enough activation energy or whether they collide in the correct orientation. We're going to go on to look at how we can model an enzyme reaction, and the first model we're going to look at is called the lock and key model. The lock and key hypothesis derives from the fact that within the tertiary structure of an enzyme there is a part of it that's complementary in shape to that of a specific substrate molecule, and we call this area the active site. This is like how only the right key will fit into a lock, and only a specific substrate will fit into the active site of an enzyme. To make this model simpler, we're going to use this representation of an enzyme. An enzyme is going to react with the substrate, and when the two join together, they form an enzyme-substrate complex. The substrate is held so that the right atom groups are close enough to react. The R groups within the active site may also interact with the substrate and form temporary bonds, which may put strain on the bonds within the substrate and help the reaction along. The substrate or substrates will then react, and the products or products are formed in an enzyme product complex. The products or product are then released, leaving the enzyme unchanged and able to take part in any further reaction. More recently, evidence from enzyme research has suggested that the active site actually changes shape slightly as the substrate enters. This addition to the model is called the induced-fit hypothesis. The initial interaction between the enzyme and the substrate is relatively weak, but these weak interactions rapidly induce changes in the enzyme's tertiary structure that strengthen the binding, and this puts strain upon the substrate molecule. This can weaken a particular bond or bonds within the substrate, and therefore lowers the activation energy to help the reaction along and the products to be produced.